All right, so what I wanted to do is talk about gene therapy in Huntington's disease. So what I have discovered really, and perhaps some of you are more sophisticated already, but what I've discovered really is over time, this has become a really complex field in Huntington's, and just understanding what exactly does gene therapy mean. So I'm going to go through some of it. I think a lot of people kind of think, well, if we could just fix the Huntington gene, and it's not quite as simple as that. And doing that's is kind of a challenge. But there's a lot more to gene therapy in Huntington's disease, and we have a lot of different research that's ongoing and trials that are ongoing here. So I'm going to step back. Oh, there we go. Okay, so this is my organization here for today. So we've got a timeline, briefly, some diagnostic features. I'll just go through some of the Huntington's features. Genetics, and then the therapy, and some novel treatments for today, okay? So Huntington's, we've come a long way. So and it's kind of a busy slide, but Huntington's disease was described over 100 years ago. 1872 was the first description by George Huntington. And it took nearly 100 years before we could figure out that this was a genetic disease and identify the gene for Huntington's disease. And this was really thanks to Nancy Wexler and her group. We went down to Venezuela and also, if you haven't read some of her works, she's an amazing woman. There's actually some specials out on PBS for her, but A Woman in Science is really actually a wonderful story. Since then, we've come a really long way. So I kind of highlight a few things. So along the way, in 1993, that's where we discovered the gene for Huntington's disease. It used to be called interesting transcript, but now we know it's the Huntington gene, and we know that the protein it makes is called Huntington, kind of linked together. And roughly around 2003, we had our first phase 3 trial, and then in 2006, the medication that many of you might know called tetrabenazine was first approved and shown and published. And now kind of the graph kind of gets scrunched together. So in the last 10 years, we've had a ton of different trials, including Ionis was the trial now called Roche Genentech, who took up the drug for their antisense. And I'll talk a little bit about that. Was the first gene therapy trial done. And since then, we have all kinds of stuff, and I can't even fit it on the slide anymore that's going on. I'll mention a few of those as we go along. So what's Huntington's disease? I probably don't need to tell you guys this. But just to summarize, we have a triad of different symptoms: motor, cognitive, we call it neurocognitive symptoms, and behavioral symptoms in Huntington's disease. So just as a summary, reminder for folks, Huntington's presents in a lot of different ways, okay? It's not the same. We talk about seeing one patient, we see one patient, every patient is a little bit different how they present. And even the motor symptoms. So we think about chorea, but chorea is kind of the tip of the iceberg. To tell you the truth, there are lots of different manifestations of Huntington's disease. So dystonia means involuntary sort of abnormal posturing. There's stiffness, rigidity. There's lots of different symptoms on there. Speech, swallow issues are really common as well. So just the tip of the iceberg. Okay. In terms of cognitive symptoms, we often see these symptoms occurring early on in disease, and they may start out really subtly and they're very insidious. So just beginning with kind of problems with organizing, thinking about things, maybe have a job, maybe having a little bit more difficulty with the job, managing things, even managing your household things, just taking care of the daily things of getting groceries, making lists, thinking about things, taking care of yourself as well. So over time, that progressively gets worse, okay? Not every patient, but many patients at the very end develop dementia, okay? But it's a very slow, insidious process throughout the course of the disease. Likewise, many patients suffer from behavioral issues or psychiatric issues. I say behavioral because it's more than just mood and depression and anxiety. It can be outbursts of anger, it can be outbursts of irritability, it can be being more apathetic, not withdrawing from things, kind of being socially isolated, to people who actually are getting in trouble with the law. So it's a complicated disorder. This is just one slide, okay? So I just wanted to summarize that. But today I'm going to focus on what causes Huntington's disease, the genetic factors, okay? So we're going to go from our chromosome all the way to this thing down below, which is our DNA coded by a 4-letter thing. And we're going to talk a little bit about it, okay, today. So took a long time, but we now have identified that the gene for Huntington's disease is located on chromosome 4, okay? And it was cloned back in 1993, as I mentioned before. The gene is a really big gene, okay? I'll come back to this, okay? So many of our genes are coded by both what we call exons, those coding portions, and then we cut out pieces of our DNA called introns. This one has 36 codons, it's huge. And the mutation for the gene is actually in the first part, okay? So the very first exon, the beginning of the gene itself. It turns out the mutation itself is a repeated sequence, okay? So our DNA is encoded by 4 letters only, and it repeats itself normally. In this case, it's a repeat of CAG, okay? We'll come back a little bit to that, but the bottom line is we see a expansion of that, okay, in the exon, the first part of the gene that leads to Huntington's disease, okay? And I just showed just a little bit of a picture on the left here, okay? You may be familiar with this when we talk about, and I think Lee will talk a little bit later about this particular thing, so I'm not going to spend a lot of time. But when we talk about risk for Huntington's disease, we talk about this repeated sequence. So often you hear about, what is my number in Huntington's disease? What is my CAG number? That refers to the mutation in the size. Everybody has 2 copies of your genome, so 1 copy of the gene from your mom and 1 copy from your dad. We call those alleles, okay? You only need 1 allele to cause disease, or 1 abnormal allele. And so normal alleles on the bottom have a copy number between 10 to 26, roughly, okay? There's an intermediate level, and then you go up on this tree. Anybody who has 36 or more copies is at risk, okay? Affected at risk for Huntington's disease, okay? Now there is this range between 36 and 39 where you have this intermediate, this reduced penetrance we call, you're at risk, but it's not a 100% chance. If you have 40 plus, you're at risk sometime in your life, 100% chance, okay? All right, so just wanted to raise that. The genetics gets kind of complicated in this sense, okay? So just because you are at risk doesn't mean always that you're 100%, and that's what this kind of, why that is relevant, okay? And you can kind of see the longer or the larger this expansion is on the right side, it's a graph showing sort of age of onset. Also, I didn't put severity there, but age of onset, you can see increases, okay, all right? You can see as you go out, age of onset decreases as you go with a longer or a larger repeat size, okay? Most, on average, patients who have a repeat size in that sort of low range, in the 40, onset of these could be anywhere from 20 to 80, okay? But as you go out, you can see increasing frequency that the age of onset gets earlier and earlier. And actually that correlates with disease severity as well. So what does this protein do? We don't really know as much about the protein in terms of its normal function. It's a huge protein, actually. It's one of our largest proteins, okay? The mutant protein we do know a lot about, okay? It fragments easily. It forms aggregates in the cell. Basically, it clumps up. So, and these clumps actually you can see on the microscope. So I show you a couple of pictures. On the left is sort of staining kind of with the fluorescence. You can see the little bright circles there, so they're listed in red. And on the right, you can kind of see there's a little brown clump in the nucleus of the cell as well, okay? So you can imagine kind of something collecting, clumping up in your own body, all right? It blocks different cell functions. And in fact, this causes a lot of problems in your cell. Starting with the nucleus, it can affect expression of different genes. We know that. In the outside of the cell, it can express, it can cause problems with the function of different proteins, okay? And even the function of some of your cell organelles, like the energy production in your cells. And some of these fragments can get out and then lead to inflammation in the brain. So there are multiple different pathways listed here. So it's a complicated slide, but complex the different things that go on in the cells. So it's almost like thinking of a cancer in your cells. It clumps up like a cancer, it kind of grows, and can get out of the cell, and it can spread its pathology to neighboring cells in the brain, and even supporting cells causing inflammation, and then causing eventually the brain cells to die, leading to the brain to progressively degenerate, okay? So this is what we know about Huntington's right now, all right? So let's come back to the genetics, because the real question for you all is how do we treat this disease, and how can we find a cure for us? So fix the gene is kind of what I always say. So it all goes back to the fact that we know there's a genetic defect in Huntington's disease. So, all right, I'm going to talk a little bit about the different structures here, because it gets all mixed up. So I already brought up DNA, I brought up protein, and then there's this other thing we call RNA. What the heck am I talking about, okay? So let's dive into this a bit. So DNA is your instruction set. So it's this 4-letter code that we talk about. We have about 20,000 plus genes in our body that encode proteins that make up who you are. And you pass these along and it makes you who you are. Someone, if you're a brother and sister, you're a little bit different. You have a different eye color, you have a different hair color, okay? That's all based on this instruction set. But how does this instruction set encode the proteins that build your body? Well, first of all, the instruction set gets copied, okay? So our cell does that, and it makes this messenger that's called RNA. Okay, we use those instructions, we copy them first, and we use that to instruct the cell to make proteins. And proteins are actually made up of little building blocks we call amino acids, okay? And those are kind of built up in this little tiny sort of string of beads, basically, and then it folds, okay? And so a folded long string of amino acids makes up a protein. And so you go from a DNA, the instruction set, you're kind of copying the recipe, you take it home, you get your ingredients, and boom, you got a cake. That's your protein, okay, in the end. Does that make sense? I hope. Okay, great. So there are all kinds of different levels at which we can target our gene therapy, okay? It's amazing promise. We got a bunch of novel treatments. We can target different levels. But if we talk about targeting at the DNA or the RNA, we can do things like increase the expression of something, or we can decrease the expression of certain proteins that might affect disease, okay? And there are different ways that mutations in our gene can affect the way proteins work, okay? So we often talk about either we call loss of function mutations, okay? So these are mutations that sort of a genetic defect. So if the recipe's wrong, you get a bad cake, right? Okay, that's a loss of function mutation, all right? So if it tells you to put the wrong ingredient in, you miss the salt, that tastes terrible, okay? Some genetic mutations actually cause a gain of function, okay? And what I mean by that, so sometimes if you take a recipe and you add a whole bunch more baking soda to your cake, suddenly you got a cake that's a lot taller, okay? And it rises more, or it might do more of something, okay? And more of something sometimes is okay, but sometimes more of something leads to other problems, okay? So too big of a cake, it'll fall over, okay? Another kind of thing is too much salt will make you a really salty cake, and everything is just gonna be ruined, okay? So sometimes you get mixed effects too. So some things you do in a recipe, you get sort of mixed effects. Some things taste worse. Some things make the cake fall over. In the same way, genetic defects can be really complicated. So Huntington's, we're still kind of out. What we think really is the mutation for Huntington's disease has kind of two roles to it, okay? First of all, it makes a dysfunctional protein. So that's a loss of function. So the protein does something and it doesn't do it anymore. The other part of Huntington's disease is that sort of we call the aggregation. The protein, the mutant protein, has this expanded portion of it, so it forms this huge sort of, we call polyglutamine, this enlarged protein. It clumps up in this cell, causes fragments, and those fragments have toxic effects. So that's a gain in sort of functional problem. So it's a complicated feature of mixed different effects, okay? So we actually have ways to manipulate this, so we can actually use this genetic information to manipulate the way the gene works too. We can edit it. We have techniques, and I'll talk to you about this new thing called CRISPR. We can introduce genes to modify other genes, okay, basically that's the gain of function stuff. We can change the way the gene works, so we can knock it down, so take a defective gene and reduce its level. Okay? And another way to do it is like doing silencing and knocking down. So that's another way too also, okay? So I'm going to talk about each of those. So the important thing I want to mention is like the different levels here. So remember, we start with the instruction set DNA, we go to the message RNA, and then on the far right we go to proteins. That's the end thing here. So actually a lot of ways where we can target our therapy. So in the beginning, if we could just fix the genetic defect, that would be the way to go. But this has become quite challenging. However, I'm going to talk a little about CRISPR, these talons, there's a lot of different things that are actually out there that are still research-based and they're still in the lab. They're yet to be efficient enough to use in patients, but it's coming, okay? So that's where I get back to, let's fix the gene. There are other things that can target the gene sort of at the nucleus level. We do have some things beginning to target the message. So there's antisense oligos. So these are actually pieces of DNA. They're really tiny pieces of DNA that we can inject. And I'll talk more about this, but this is an example of this is the Roche Genentech gene therapy uses antisense oligos. So short pieces of DNA that we can inject into the fluid around the brain. It'll bind to the message and knock it down, okay? We have other examples. I'm going to talk briefly about using a virus to express a RNAi, we call it interference, where you can also, again, knock down the gene. Once we get past the message, we get to sort of this early protein. We have therapies looking at antibodies. So antibodies are natural things your body produces, but we can make them in the lab. So we give you vaccines. Vaccines tell your body to actively make antibodies to things like viruses, like the COVID-19 virus. We can also generate these antibodies in the lab and just inject them into your body passively, okay? Not the easiest thing to do, but we can do that. And we can sort of lower proteins that are problematic, okay? Like the mutant Huntington protein, okay? Targeting is the issue, okay, with antibodies. Okay? There are other things like Vertex that target the sort of aggregated proteins, so helping to degrade them. And then, of course, there are other medications that target proteins in the cell that may have effects, indirect effects on the mutant Huntington protein to either lower it or remove it or get rid of it so it doesn't cause its effects on the brain cells. So a lot of different levels, okay? So just take a step backwards. Again, I go back to Clinton, it's like the economy is stupid, it's the gene kind of stupid, that's why I think of it. Ultimately our genetic researchers tell me, it's great all this stuff, but if we could fix the DNA mutation, that would fix everything. So it sounds great that we can do this. The problem is, every single one of our cells has to be fixed. The Huntington mutation is in all of our cells, okay? Not just in our brain. So we have to get to each one of those cell nuclei to fix that. So we need an efficient way. We don't have that yet, but we do have ways, I'm going to talk briefly about it, of editing the genome. So this is actually really new stuff, okay? So in the lab we use CRISPR, and you may have heard of CRISPR. So CRISPR is this new technique. We also have a few other things that are listed on your talons. There's zinc fingers. All of these are just molecular mechanisms that we have just developed in the last 10 years, okay? To go into the cell nucleus and actually edit the genome. It's a little bit scary, to think that you could actually edit someone's genes. You start to think about the opposite side. So fixing a disease is great, but editing someone's genome, or let's say at the embryonic level, to make somebody have brown eyes, blue eyes, or make them smarter, or or greater, or select out something that you just don't want. So there is a potential ethical issue here that the whole world has already struggled with. There actually have been at least a few examples. One example in China of having a CRISPR-edited baby being born. So now that is outlawed. Technically, you cannot do that in the United States. However, there is the potential here for editing people, and we do use some of these techniques or we're trying to use some of these techniques in the lab to treat certain diseases, right? So, but it does have its side. The way CRISPR is kind of complicated. It's just a piece of RNA that we make, and it can get into the actual DNA. Nowadays, it's not just that you can stop a gene, you can actually introduce a sequence. So for Huntington's, you have this expanded sequence, you have to edit it down. You don't want to really delete it, okay? So we have to do more. So one of the big questions about gene therapy is that, how do you give it, okay? Unfortunately, you can't put it in a pill yet, although we're getting there. Delivering gene therapy to the brain is a big issue. The brain has a blood-brain barrier, and even when we have drugs that affect the brain, they have to get past that barrier. okay? So we have to design, all the drug companies have to design drugs that can get to the brain. Delivering drugs that have DNA or genetic modifying effects is difficult, okay? They're not something you can just take in a pill because they'll get degraded by your gut and your stomach and all that acid will never get to the brain. So as of now, you've probably heard that many of these gene therapy trials require you to come in and either have something injected into your spinal fluid on a regular basis. That was the Roche Genentech study. That's just one example. Other ones are actually the brain. But we're looking at different methods of doing this. So, but antisense is one way, but delivery I like to talk about. And now there are different ways of doing this. We can actually use viruses. So I know it sounds scary to take a virus and use that and give you a virus. But the truth is that we have actually engineered some viruses that work really well to infect, well, I wouldn't say infect, but get into the body. So a couple of examples are what's called the adenovirus. Adenovirus is a common cold virus, okay? We've engineered it, so it's called, the one that we use is called an adeno-associated virus. So it's engineered not to cause disease. But we can use the virus because it's a natural machine. So what viruses do is they carry genetic information, and they put it in the cell, and they use the cell to kind of replicate them, and that's what causes disease usually. So, you know, many decades ago, we discovered we could use these viral machines, quote unquote, make them so they don't cause disease, but use them to introduce genetic information into different tissues. So AAV was actually something developed here at the University of Florida, so we're pretty proud about it. And the technology has really gotten much worse. So, and there are other viruses that actually do that as well. They're really great. They're natural, they're really efficient, they're stable over time, and the engineered ones, you know, again, don't cause inflammation or disease. So that's why I'm kind of pointing out on this particular slide. They don't make you sick. That's one major factor here, okay? One key thing here is they do have a little bit of a relatively small package, but we still can pack a lot of stuff in them, okay? They're easily produced. In fact, in the lab, we do this routinely. We have the PAL gene therapy core at the University of Florida here, and they can make enormously high copy numbers of this. And the PAL gene therapy core actually has produced gene therapies for certain diseases, okay? Not yet for Huntington's disease, but I think coming, okay? There are some advantages. So I talked about the advantages. I didn't say about the disadvantages, actually. We want to make sure we use a virus to introduce a gene that we don't have. We want good expression. We want to make sure that the effects are targeted. Side effects for any protein is basically what we call off-target stuff. So we want to avoid side effects. One thing about gene therapy, you know, if you mess up gene therapy, you could potentially cause something like a cancer. Okay, some off-target effects that you don't want to happen. Okay, so you have to be really careful. Okay, but any therapy you have has to be easy to produce. Okay, it has to be effective, has to be targeted, have low side effects. Okay, and those are key things when we develop any kind of medical therapy. Okay, if it's not all those things, then it becomes really expensive and it doesn't really work for patients. So this is just where we go with therapies. I'm going to finish with this slide, basically. So we have a lot of different therapies. We're a lot of stuff is up here in the preclinical phase, which means we're still at the bench. So CRISPR is a lot at the bench, okay? But we do have other therapies that are moving into phase 1 trials. This is where we try them out in a few patients. And as we go to the right, we do it with larger and larger numbers of patients, and we're looking more at, do these things work? So we do have different trials that are going on here. So this trial called Unicure is ongoing. It's actually now in a phase 2 trial. This is a trial where we actually use a virus to knock down the Huntington protein. It has to be injected into brain, okay? So it's amazing. The promise there is the delivery could be a one-time delivery of a therapy that might slow the disease down. So huge promises. The question is about risk. It has been stopped for a couple of safety issues, but I think it will probably go forward. We take 2 steps and step back always with our research. I hate to say that, but that's just the way research goes. A couple steps forward, steps back. Both Wave and Genentech did the antisense trials, and if you haven't heard, I think in the last year, they came out, they had to stop the trials because of what was called futility. So
They were amazing. They enrolled over 600 people worldwide, and treated patients. We were able to show that we could sort of extend the sort of injections. They were injecting therapy into the spine, and they were doing it every 4 weeks. They got it all the way out to 16 weeks. So not as frequent, okay, but it would be lifelong, every 16 weeks of therapy. A lot of hopes were tied into these trials, and they didn't show any benefit. Okay, so they were stopped for futility. So this is where we take a step back and say, okay, what did we do wrong? Was it targeting? Where do we go next? And that's kind of where we're at the moment. Roche and Genentech are thinking we needed to start earlier, so earlier patients. So we don't want to target people who already have Huntington's and are more moderate to severe. We want to get really early patients. They're planning on doing another trial. So there'll be more on that soon. I haven't got that on this particular slide. I said there's no pills yet. Novartis has a trial called Vibrant HD on a drug called branaplam. So it is marketed as a gene therapy, but it is a pill. So it's an exciting development coming through. It's still not widely available around the country, but there's some excitement regarding that. So we're not giving up on this gene therapy idea, and there's still other ways to find delivery. So that's where we're at. You see a few other trials listed on here. Proof, there's Connect. These are actually treatment trials and not gene therapy. Okay, so try and treat things. So, but they're really great trials as well. They're ongoing. A lot of people are enrolled in them. And then, of course, eventually we want to get to treatment. Okay. So, all right, I'll finish. Multiple things for Huntington's disease. Going a lot of ways to target it. Gene therapy means a lot of different things. So where do we target? From the gene or the protein, or somewhere in between? All of them actually will help Huntington's disease. There are a lot of hurdles to have. These viral vectors are really cool, very promising. AV science, the science of CRISPR is rapidly expanding. I think of it like computer chips in a way, you know. Remember, we always started out low and then sort of this exponential curve of sort of advancement. And I think that's what's happening with molecular science. It's a little bit frightening sometimes, but and we're taking steps. Some things go in spurts. Okay? New clinical trials are definitely coming. I'll stop there. Go through.